So another thing, Ben, I'm told that where you left the marker at the top, it's a place where ROVs go to die.
Oh, let me
That's
let me
that
just
gets
wear
up
that
in the
marker
fishing
forever.
gear.
Okay. Yeah, I think that's kind of what we're trying to avoid. So it's good to know. Thank you.
So I think Lizzie put the Z4 there. It's kind of like, okay, maybe go up about here, but let's not keep going too far past that.
Okay, yeah, no, I think the goal will just be to keep meandering along this for quite a while, and we can get back up on that ridge. But the farther we go, then the safer for us. So that's good to know. Thank you.
Yeah.
Place where AUVs die. AUV graveyard.
If you need any spare parts, it might be a good place to go.
God. I don't think it's worth it.
No.
And Chris, are we going to be running a transect along this contour as well?
Yeah, I'm starting to think we might as well. If you feel like you can maintain sort of a semi-consistent flow based on the terrain, I think we might as well go ahead and get one in.
Sure thing.
Yeah, I think
we can start that whenever you guys are ready.
Raj.
Yeah, I'm going to keep the 110 coming in. And
110, Raj.
Yeah, you can just go up and down if you
confirmation on this is where electronics go to die. Kurt is typing in that at the pinnacle at the top has a fisheries service camera sled sitting at the base.
Okay.
Nice.
It's going to remain there.
All right, Ben. I'm going to start my move.
Yep, I'm going to call this now.
Roger that.
Yep. Transect 10. Bridge nav, you can add.
Are lasers on, or are they just blending into the sediment?
Oh, negative. They are not on. Now they are.
Good catch.
Mary, you are on top of your game.
Data eyes.
I'm usually bad at finding lasers when I do video, so can never tell if it's me or they're not on.
I think you're right, Kurt. I think one of the Navy ROVs was able to get it back for the Northwest Center, but it had been on the bottom of the ocean for several years by that point.
Well, so all their anodes are probably gone.
I would guess, yes.
So is that area still open to fishing, or it was historically and it's no longer?
Yeah, historically it was, but now it's no take marine reserve. That said, it's very difficult to demarcate boundaries for these things in the ocean, and so it's not necessarily clear to just the lay fisherman who wants to go out and throw a line for fun, for recreation. So we have a lot to do. One of our largest tasks is education, getting out there and letting the folks know in the water what areas are okay to fish in and where to stay out of.
Yeah, I bet that's difficult.
But it's not, because it's up where we are, very close to Anacapa. That's also that's the easternmost island, and it's also the therefore the closest to land, to Ventura and Santa Barbara.
Right.
And so it's it's one of the more easily accessed places. So we get a lot of fishing there. Could we get a zoom on that? Oh no, sorry,
Negative.
we're on transect. But all of a sudden we see something exciting I want to check out. That was cool.
Always happens when we start the transect.
Yeah.
Something cool comes out.
I'm jumping. So the westernmost islands it tends to be a lot more rough out there in terms of weather conditions and oceanographic sea state, and then and it's further from the human population. So footprint is an appealing spot. If you look on a map, you think, oh, look at this little bump. It's a great spot. I can go out there and look for some rockfish. But it is currently a no-take reserve.
Yeah, the ocean's a big place to manage, but it feels that way.
We've found with our AUV that footprint has lots of little nooks and crannies for robots to get stuck in.
Oh yeah. Even regardless of the fishing gear.
Was that an actual study?
That's just a bonus. The fishing gear challenge.
Oh man. We'll have a RV made out of PVC to do that course.
We've been able to do quite a bit of work in the sanctuary using the sanctuary vessel. And we did have a study down here where we compared an AUV, an ROV, and a manned submersible
Oh wow. That's an interesting one. Really interesting.
in the data that they collect. And I don't think there was a clear winner.
Yeah, I bet they're all just compliments to each other. The AUV being so like rigorous and replicatable. The RV being like finer scale stuff. Is that fish? Okay. What's he doing?
Oh, look at that.
I think that one's a calcod.
He's big.
I think that big one was a calcod that we just went by.
So Erica, have you been able to do work in submersibles yourself?
I have not, and I'm not sure that I would, given the opportunity.
Yeah,
Just knowing how pressure works.
I know, right? It's like Das Boot. U-boats. I TA'd one semester, missed one research cruise, and both my lab mates and my advisor both went in Alvin on that cruise. I was like, oh man, I missed something exciting. But yeah, the concept is definitely spooky too. Be neat to see though.
Always putting in a plug for shipwrecks. Bob says that some of the best sport fishing locations are on the shipwrecks around the islands, providing hard structure or habitat. And that is often true. Shipwrecks or other man-made things are also often a spot where we find invasive species, new hard bottom and structure for them to colonize. Pier pilings and along the whale rigs too. We have an invasive bryozoan that's been working its way throughout Southern California.
That's interesting.
A couple other invasives in the shallow water environment, two kelp species, Sargassum and Undaria.
Roger.
Undaria is the algae that you find in miso soup. Both are from the other side of the Pacific Ocean, however. And locally, we worry about Sargassum, which recruits to hard bottom habitats, competing with the kelp, the Macrocystis giant kelp.
Looks like some Phorea there. Definitely spongier over here.
With how little we know about the deep sea environment, it's very hard to tell if something is non-native or just we haven't seen it before.
And I have not been given the opportunity to dive in the delta, but I know that Tom has.
Oh, yeah?
And his lab, they used to work quite a bit with delta.
How deep does that go?
I don't know about that one. That one's before my time in the
Just a
robot
giant
game.
metal bell? No.
It could be. And then his lab has also worked with the deep worker and the dual deep worker. There's another salad bowl sponge.
Very newly described earlier this dive.
I'm glad Abby's here, so she knows what my notes will mean when I say salad bowl sponge.
How could she not? So aptly named.
I don't know, this whole wanting to call everything by Latin names and by rules, I don't understand.
Yeah, I agree.
There's a mini salad bowl. Bank rockfish.
Oh, yeah.
So Abby, do we think those shelf sponges are something we want to keep an eye out for later? Yes, please.
Jess is keeping in a constant altitude from the seafloor and the seawall.
Nope, this is great.
Cool.
I think that's another bank rockfish. Lots of fun sponges for dye to identify.
You just have 50 meters left in the transect.
Raj.
Another big calcod.
Yeah. Salad bowl in the distance. Finally.
So are the salad bowls common enough now, Abby? Are you feeling better about it? larger Sebastomas up under that ledge.
After 30 days.
Some Parea. That was quite the spongy slope. That, I think, was your Picacio.
And how much further in this transect?
Just 10 meters.
Roger.
Right before another little spongy slope. Hopefully spongy.
Keep an eye out for a salad bowl.
Okay. 3, 2. All right, that looks good. Let's mark it.
Roger that.
Great job, Jess.
Is that a salad bowl just up there? Yeah.
Where are you looking there, Chris?
Actually, I'm on a delayed stream. I think we passed it. The
There
regular
was
stream
one
was
up to the
stuttering
left.
on me.
Yes.
We'd have to back the ship up in
Yeah,
order
do
to
we
go
want
that
to
way.
explore around here for a little? Or
That'd be great. I'm sure there's another one
Yeah.
if you are moving in a general direction already.
Sure thing. Oh, is that one here?
ooh, looks like there might be one on that rock to the bottom right too.
It looks like there might be like 3 candidates here.
Not a salad bowl expert though.
There's enough salad to go
Or
around,
a
you guys.
pasta
Don't
bowl
worry.
expert.
It looks like there's potentially 2. Are those 2 salad bowls to the left of the lasers or one down to the right?
Yep.
Lower right?
Which I think they're all the same, so whichever
Right.
one you think you would have better access to for a little grab.
Yeah, sure.
Did we also want to do a Niskin for Meredith?
Marley, are you full wide there?
Yeah, I think so. Yes, yeah, basically.
Great.
Mary, you want a Niskin?
I think Meredith said it in the chat, but
Look at that decorated crab going in.
Yeah, can we get some, she would like some zooms to start.
Sure. 1 sec. I'm just gonna settle out real quick.
Okay.
Bulldoze these ones in the process.
And she's thinking that little one right under the lasers now might be the best one to sample,
Baby bowl.
the baby bowl. But if we could look at the others too.
So I'm gonna land right here. All right, Ben. All right.
Pretty stable.
All right, Marley, go ahead and push on in. Is there anyone in particular you want to zoom on, or just the cluster of 3?
I think both that one and then the one down below it
Roger that.
when you get a chance. So
Raj.
inside and outer surfaces, please.
Sure. Sure thing.
So I'll just zoom all the way in on the middle guy here,
Sure. Well, I get you the lit one here. Has better light. Oh, come wide there, Marley. Don't wipe you guys all over the planet here. All right, go ahead. Okay, I'm gonna. That's all I have on the. Let's do one quick bump up.
What if you rack back a tiny bit? Will that trick work? Oop.
Yeah, like this.
Yeah.
Try that out.
Yeah.
That full zoomer.
Yeah, that's all the way in.
Great.
Very nice. Thank you.
And then you want the outer texture as well?
Yes, please.
Marley, you
This
actually
is
can
Abby.
push back in.
Yeah.
This is Abby saying it, so imagine the British accent.
you don't want to give us your best British accent.
Not with Abby listening.
All right. Exposure and focus on these is a little tough. I think the outside is in focus. Hopefully.
Funny, I was just thinking, how do you focus on something that's fuzzy?
Yeah, well, the problem with white sponges and light brown sand in the background is my focal thing just doesn't really show any difference between them. But that looks good enough, I guess.
Yeah. All right, and then we'll also look at the bottom guy here too.
Cool.
You want to come a little wide so don't
Yeah.
you guys sick?
The victim.
My victim will be this little guy. Get Marley, push on in, please. Do I dare try to center it? Ooh, it's gonna be hot.
There.
Oh, dare too far. Good enough.
Do you think that's the same species all around it, the white sponge material?
I think that one's different.
Oh, okay.
To the left, that looks almost heterocone-like.
All good there, Science?
Is that the one we want?
Yep,
Abby?
that's be the one. Oh, sorry.
No, I'm just making sure. Okay, so she's agreeing with me. Yes, that is the one we want.
Sweet.
Sample
Roger.
away. And Abby, are you thinking grab? She's thinking grab.
We can grab, Roger. And Mary, what do we got open that starboard bio box of ours?
We have A and B.
A and B, Raj.
It's going to be 122. Sound right?
This guy's pretty itty bitty. Do you want a haircut or do you want just the whole shebang? Kind of voting for whole shebang.
I would say whole shebang.
Roger that.
Shebang it is.
All right, Marley, here we go. Gonna push on in.
Sure thing.
That's good. My jaws
I
are
think Abby
creeping
would be more conservative, but it seems easier to grab the whole shebang.
Yeah. It might be a little shebang
A little shebang. Whereas Tom would say he wants the top left-hand lobe
Right.
on the far side.
again.
And could you please cut it so it's 1 inch square?
A millimeter deep.
I don't think this is complicated enough for Tom. He would want to challenge Jess.
You could just blindfold her.
Oh dear,
No,
that's setting us up for some really scary imagery.
That's right, he wanted to turn the lights out this morning, didn't he? Abby, do you have some other description I should put on this other than salad bowl?
This would not fit
What?
a salad, by the way. This one would hold just a handful of nuts, maybe.
Or your
The candy
peas?
dish.
Or your keys, yeah. Or like, yeah, a couple pieces of candy.
I was thinking egg cup, but
Oh, yeah.
There's no going back now, guys. Oh,
nice.
wow.
All right,
Nicely
Marley,
done.
go ahead and come wide, please.
Perfect.
Abby likes the description.
Abby says to
salad
put white
bowl.
funnel.
White funnel.
All right. Go ahead. I am ready for ya. Let's go for B.
Nice job, Jess.
Sweet.
Now it's low grip force, so I need to get my hand out of the box. There we go.
Nice zoom on some dirty barrels and a faria on the distant rock.
Chris or Erica, did you want to fire a Niskin for Meredith?
I haven't seen many corals.
I
We are
thought
at the end
she-
of a transect, but we are at the end of a transect, so we've only done 2 so far.
Correct.
Yeah, I would say go ahead.
Roger that.
All right.
Dun, dun. Kim, you want to get some eyes on the Niskins there?
Right.
Mary, we're gonna go for number 3. Is that correct?
Correct.
Oh, on the port rail cam there. Sorry, Kim. I should have been more specific.
So this is NA123 sample 123.
Ooh,
I know, very exciting.
11.
Guys ready?
Oh.
Yeah.
Got it. 1, 2, 3, 1, 2, 3.
And Niskin 3.
And Niskin 3. Meaning we've gone 1, 2, 3 on that. Moments like these are rare, guys.
Thanks, A-Team.
All right. T. Carry on our merry way.
Yep. If
Remind me
Science
the
is
merry
good.
way.
yeah, so I was just gonna say, I think I'm gonna change this to 100.
100, Reg.
Is that good, Science? Keep going.
Yeah.
That's good for this spot. Yeah, I think we're still looking for some of those flatter plate sponges.
Roger. Yeah, we can
So
explore around.
Yeah. Okay. Does that rock? Looks like it's
So didn't we collect one of those platy sponges this morning? And Abby wants
we did.
yeah, so she wants another to compare.
If the salad bowl can have, okay, so now we're discussing whether the salad bowl can have that plate morphology or if those flatter ones are different.
Oh, that's interesting.
So we did collect one this morning, but there was a different, a more upright plate in that same area that was gray and white. It seemed to have a grayish tint on the outside. So then if some of these, if the salad bowl, it does look like that it could be two different morphologies of the same sponge.
Interesting.
And a beer bottle.
Message for her. It's like a big rockfish again.
Oh, look at him. That is, it's another cowcod, I think.
That rockfish is like, if I stay perfectly still, they won't see me.
So wait, all these big guys we're seeing, you call them cow cods? Cow cods.
Cowcods.
And it's
Yes.
not a cod. It's
Can we get a zoom on him, maybe?
Oh, yeah.
They have a
Oh,
really
wow.
pointy face, long pointy face.
What?
The dorsal spines are deeply indented on their dorsal fin. They have alternating bands of light and dark,
Super.
and they get pretty big.
His spine's like on his operculum too.
Yep, lots of spines. That's how you can tell some of the rockfish. You count the head spines.
I like that, deeply indented. I'm like, oh, okay, I see
Ooh,
what that means.
could you zoom again on that yellow? Is that a coral? Right in the center of that rock? No, Kurt, are you still here? Do you know this coral?
And Daniel's asking if we could zoom in on the white spots by the tail of that fish while we're here.
Yeah, I'll have to bring the.
Or we passed it.
Yeah, we just. The big cod rockfish we were looking at, is that they want to look at the caudal fin or something? Oh, you're
Yeah, just the bottom.
yeah, Chris, is it
It's
still that same fish that we want? The caudal part?
We actually just want to look at the mollusks on that are next to that fish, just below, like at the base of this rock here.
Roger. Can we interview?
Yeah. Daniel's wondering if there might be brachiopods in that mix.
Oh, Raj wasn't on SPO. That would make a lot more sense. You guys just want this here, or was there anything else you wanted to see?
Yeah, I think that's where he wanted to look.
Don't see any.
And Jess, if you, yeah,
He says,
when we're done with this, if you could do another flyby that coral, the
Sure
yellow
thing.
coral, that'd be great.
Roger that.
Good.
You good here, Chris?
he says, how close can we get in terms of a zoom?
I'm not in, I'm like halfway in. I can, if there's a, is there a specific spot that you guys want to get a full zoom on, or just any of these?
I think about, yeah, I think about where you're looking right now.
All right, here we go.
Zoom, baby.
That's full zoom.
Raj.
Yes, Di, the rockfish is a cowcod. But we're going to pan to a coral here in a second that I was hoping you could help me out with.
All good there, Chris?
Let's see. Daniel's asking for anything else. Yeah, I think we can zoom out.
All right, come on, Marley.
Yeah, we're good. Thank you.
All right, Marley, go ahead and push on in there, please.
I'm trying to decide if that's a Swiftia, a yellow Swiftia,
Come wide, Marley.
or an Acanthogorgia.
All right, Marley, go ahead and push on in there again, please.
Doesn't have the typical fan shape, so I'm thinking it might be the yellow Swiftia.
Come wide, Marley.
Okay, we can be done with that one. Thank you.
Roger. Anything else you guys want to see around here, or else we're going to start moving again?
Nothing on this end. Erica?
Nope.
I think we're okay to move.
Oh,
Just keep-
sorry, I didn't hear that.
Yeah.
I think we're okay to move
We're
to
good.
keep
Roger that.
exploring around.
Right, Ben. 100 still. Raj. Ben, can you zoom out on HIPAC too? Just see where we're at. Thank you.
Hello, Meredith. Can you rewind about 5 minutes and take a look at the yellow coral we just looked at? Oh, here's another one on top of this rock.
Roger, 105, Raj.
Is that possible to quickly zoom as we cruise by this at the yellow coral on top?
Sure thing, Science. All right, Riley, go ahead and do a snap zoom, please. Thank you.
They really stand out. Thank you.
Awesome. Go ahead and come in, Riley.
Is that something we want to collect, Meredith, or are you good without?
Should I sit down here, Science?
I'm waiting for a response.
Roger.
Meredith, is that something we want?
Yeah, the ship should be fully stopped now. So
Roger
Oh,
that.
thank you.
I think we'll be seeing, if she doesn't get back, I think we'll be seeing a bunch more. I can't,
Okay,
as we go up. So,
fair enough.
yeah, well, the ship stopped.
Yeah, so is that a continue on?
That is a
yeah,
continuation.
Okay, great.
yeah.
Chris promises there will be more.
My personal guarantee.
Pinky promise.
That's a dangerous thing to make in these environments in that situation. What's the fish to the left, Erica? We just went by it.
That, I don't know. I didn't recognize that one. Why did you see
10530
that?
meters, Raj.
Yeah, thanks.
Is this the fish you're talking about, Chris?
Yeah,
The large one.
Yeah,
off to
nicely
the
done.
left, yeah.
nicely done.
Is it a vermilion?
Good.
Don't know if it's worth the chase.
I like the orange.
Orange is cool, yeah.
Yeah. Orange is cool.
Color. Good color for a fish.
Red is cool too.
We saw tons of vermilion at San Miguel the other day.
Yeah.
Vermilion and green stripe.
Or a sunset. I was wondering if it was a sunset because of how deep we are.
Okay. Can you tell them apart on video, or you need to have them in hand?
I couldn't. I don't know if Di can. I like her term though, vermilion-ish.
Nice, Di. Thanks.
That's all you need.
She's saying I don't think you can tell them apart visually. It's one of those where they hang out and then genetics. So they are depth differentiated.
Okay.
Vermilions tend to stay shallower and the sunsets are deeper.
I wonder if that type of zonation changes if, like, the oxygen environment or something changes. Or do you think it's purely just depth, pressure, light?
Oh, and Meredith has gone the extra step of saying it's debatable whether you can tell them apart genetically.
Who can we trust?
Right?
Just one of those things you know.
Well, if you can't tell them apart genetically, are they really different species?
That would be the question. Are we going to have a separation
Di
that
says,
comes back together?
But it's happened before.
How old are you getting them, Di? Di says she's aging them. The oldest one, 50. That she's aged so far. So presumably older than that.
The vermilion rockfish.
Yeah.
So the way Di is aging these fish, I'm guessing, is fish have ear bones called otoliths that keep them upright. They're just like our ear bones. They provide balance in the water column.